the last week class you have understood and everybody have received the notes. Today, like the last class of we had discussed about, I think we had discussed about chloroplast, that is plastids. So today also we are going to discuss about something that is very much important, like plant, how plastids are important to plants. Same way we are going to discuss about one of the cell organelle, which is known as mitochondria. You can see the diagram picture of mitochondria which was given there. It is a double layer membrane, and it is having some folds inside it. One of the membrane is folded, and why it is known why it is this mitochondria is very much important. This mitochondria is very much important because mitochondria is the site inside the eukaryotic cell where energy is synthesized. Because of this reason, the energy synthesized, that energy is synthesized in the eukaryotic cell as in the form of ATP. Because of this reason, this mitochondria is called as the powerhouse of the cell. And I have given some historical part of this, because all the biological or cell organelles and systems which is present over on this earth, but there will be some people, those will be uh, done some studies behind that. For the first person to report about the presence of mitochondria is Richard Altman, and he called it as bioblast. And the term mitochondria was coined by the person known as Carl Bender. And in there is one in the modern times, if we want to see mitochondria, we will use some stains means chemicals that is used to give color to a biological thing or a biological substance. They are known as stains, and mitochondria can be viewed under the microscope through a special stain known as Janus green. And this was first that combination of chemical was first made by Linear Michelis. After that, observation in the plant cell, that the first mitochondria in the plant cell, it is first by Frederick Maves. And the powerhouse of the cell, that particular term was given by Philip Shakewitz. And if you see this, and there is some more thing, ATP, that is energy coin. ATP was first discovered by Carl Lohmann and Chandrastik independently, and one of the Indian scientists also, Yelaparda Subba Rao. I think he's from Andhra. So he also contributed for the discovery of ATP. That means to find out the structure of ATP. And if the mitochondria's outer covering is not there, it is known as mitoplast. When we go into the mitochondria, you can see something, that is, see this structure. And if you see that these mitochondria are having the outer membrane and the inner membrane is folded, that particular fold is called as cristae. And inside this cristae you can see some lollipop structures, which is known as F0 F1 factors. Those are the area where energy is synthesized. And mitochondria is also a semi-autonomous cell organelle, because this also is having DNA of its own. Since it is having DNA, it is also having ribosomes. Chloroplast is also having its own DNA. Mitochondria is also have its own DNA. So it is known as semi-autonomous cell organelles. That means they are not 100% depend upon the central nucleus for instruction. Some instructions are stored inside the cell organelle itself, and there's a space between the inner space. These are, and this inside of this outer inner membrane, wherever that crystals are present, their matrix are present. Matrix means the fluid part where many of the reactions takes place, like Krebs cycle and all, which is one of the cycle which leads to the production of ATP synthesis. And the next cell organelle we are going to discuss is vacuoles. The term vacuoles means empty space. It is also a membrane structure. This is the structure of a vacuole. And if you see the historical point of vacuole, the first person to discuss about this is a Spallanzani contractile vacuole, which we'll see in some of the lower forms of organisms like paramecium, etc. Then De Vries is a person who said that the outer membrane of the vacuole is known as tonoplast. So word term vacuole means empty space, and this vacuole is present in animal cell, plant cell, and in bacterial cell. And that bacterial cell, not all the times, few time whenever it is required. And when we're thinking about the vacuoles in plant cell and animal cell, plant cells are having very big vacuole and animal cells are having small vacuole, or we can say that there is no vacuole at all, like that we can say. These vacuoles are having a lot of functions. These vacuoles can store inorganic food materials, organic food materials. It can store water. It can carry some important proteins, structures, protein structures. It can function as a membrane vesicles, many things. And these vacuoles can also act in the process of autophagy. That means cell eating. Sometimes whenever the cell is undergone some problem, this will come and eat. That particular function is known as autophagy, and this was first reported to the biological community by a person known as Emily Mechnikov. And these vacuoles, modified vacuoles, are very much seen inside the germinating seeds, where they can store some food materials for this. And other thing, we have given some differences between plant cell, animal cell, and bacterial cell. Their diagrams are also important. These cell organelles are also important. So please go through this, and if there is any doubt, comment me back wherever I am posting the video. By this, cell organelle part is over. I hope all the cell organelles I have covered. And the next part that is coming is cell cycle. That means how one cell divide into another. So before entering that, if there is any doubt inside this, please put as a comment below wherever I am posting this video.